Thank you, brother. Have fun.
Am I good there? All right. Whew. Whew. How do I pick it up from there? Well, I did need to start that, to me, this is a homecoming. If I get a little emotional, like I am now, bear with me, but it's a good way to start, right? So I just want to say that I've been here for a long time, and seeing Mike Schott, where's Mike Schott, out in the audience, just, man, it flooded me with emotions, because this place was the first place where I was able to identify myself as a scientist. And, sorry, hopefully you can hear me there. And hopefully I've been able to pass it on to some folks. So bear with me. So I've been here for, like I said, a long time. I'm not going to tell you how long I've been here. Yikes, long time, all right? So the title of my talk is Raising Fish for Research and Conservation Purposes. And I got to start literally growing larvae here at the Cabrillo Marine Aquarium, and it truly was inspiring to me. And I was able to work with the team here to grow white sea bass. And this was like my first project here as a newbie, and it was wonderful because I got to collect eggs from different research institutions, see them hatch out, and feed them to grow into various life stages. And this just thrilled me. This engaged me. This gave me a lot of purpose. Again, what allowed me to identify myself as a scientist. And people just kept pushing me. You can do it. You can do it. You can be a scientist. And it was one day when I saw this video in a research talk that really, really brightened up, my research interest in this field. So over on the right here, you'll see this fish. It's paused now because it's going to go in slow motion. And you see a smaller fish on the left. The smaller fish on the left's not so lucky. But if you see what happens, to me, I was like, whoa, I want to study this. This is what, I remember being at a research talk as an undergrad, just being like, this is what I want to do. This is what I want to study. Fish feeding. Fish feeding was it just blew my mind, right? So this is a sling jaw wrasse, and it can literally sling its jaws and capture those prey. But again, I was also thrilled by this, and it was CMA that allowed me to start my endeavors in studying how baby fish feed. So for those of you guys who are not familiar, fish undergo metamorphosis. So in this video, this is a video that really shows fish changing as they grow and develop. And I love this video. One of the things I want you to keep an eye out is for its size. So this is a time lapse showing as this little fish grows up into a juvenile. You can see its body begin to change. It's getting a little bit deeper. Its mouth is getting a little bit wider. And its fins just change throughout life. They're going metamorphosis. But look at its eye. Something's happening there. Its right eye is literally rolling over to the left side of its face, right? And I remember studying this and being like, wow, how are these fish doing this? What are the requirements for fish to grow? All of those things, and ultimately revolves feeding. So fish undergo metamorphosis. This is the southern flounder. It's a flatfish. They start off like regular fish, but they undergo this drastic change as they become juveniles, ultimately becoming an adult. I have to show you one more. I think these things are so cool. Here's another one. Here's the bamboo sole, another flatfish, but this one changes its eyes. Its eyes roll over early on in life. But again, you see that metamorphosis happening in this time-lapse video. So it takes about a month for this developmental process to happen. And we were able to observe these things here in the aquatic nursery. But again, I wanted to learn, how can I apply this to research? How can I apply this to fulfill some educational and ultimately a career? So we started off by raising fish. That's, again, the title of my talk, Raising Fish. And one of the things about fish is even though they start off really tiny, they need to eat. And fish, start off smaller than a grain of rice for most cases, and they need to eat live food. So these tiny animals are in fact predators. So this was really interesting to me, and I'm going to talk to you guys about how this interest led to 3 projects of mine. First, starting off with a project where I investigated in some larval grunion, zebrafish, and then ultimately a giant sea bass, okay? The zebrafish is the only freshwater species that I study. We'll give that one a pass. It is Cabrillo Marine Aquarium after all. But even the zebrafish serves as a huge purpose in what I was doing because it is a fish model. In other words, larval fish, many of them start off behaving the same. So we can learn a lot from studying various different species. To most of us, these other small larval fish may not look like much, but they do grow up to become very important recreational fishes. And not only that, but they have a very important ecology and ecological role in the world. If this doesn't impress you, to me, this impressed me. I sacrificed myself to take these pictures to remind us why it's important to grow fish, right? So just reminding us all. So yes, it's important to raise fish to study for science and conservation purposes. We can start off with the California grunion. This is our first fish. I probably am speaking to people who know about this, but for those of you guys who are online, the California grunion is a really unique fish, iconic. It is part of CMA's logo, if you're not aware of that, right? And its range is typically from San Francisco all the way to Baja California, and they're known for their amazing spawning season. Bear with me on this video because these videos are taken at night. At night, during the spring high tides, these fish will spawn on certain nights, and what you can kind of see there is there's a female burying herself in the sand with a bunch of males wrapping around her, and the male will give his minute contribution and fertilize the eggs in the sand, okay? And then when they're done doing their thing, you can see here, there's a bunch of eggs that have been deposited in the sand, and both these fish will return with the upcoming waves, and they'll be able to spawn again in the future. But what I was interested in is how these fish incubate and hatch. So they do incubate in the sand, they stay out of water, and then they develop within the sand and they become competent to hatch as early as 8 days after they were fertilized on the sand. Okay? But if once they're competent to hatch, you put them in seawater, shake them around, and it's like, it can be instant, right? So there's a little video of these eggs, and you can see them these little fish have started to hatch, and more of them will continue to hatch. So their hatching cue depends on receiving this wave action, right, after they've been deposited and have incubated in the sand for at least 8 days. When they do this is typically during their spawning season. Here I have from June to July. I'm going to plot some tidal heights here. And what I want you to really notice is the maximum, as high as the tide gets. So that's as high as the tide gets in any given day during the span of this month. There's my cursor. So from June to July, that's as high as the tide gets, and it is based on the lunar calendar, the lunar phase. What's really fascinating is the grunions somehow know, still not fully understood how they know, that they need to spawn shortly after these high tides. So they'll spawn at this given night, for example. Their eggs will be buried in the sand at this highest tide, and then they'll incubate and hatch anywhere from 8 to 14 days later when they receive the wave action, 2 weeks later, about approximately 2 weeks later. And this is what we call normal incubation. But what grunion possess is they possess this unique ability to extend incubation. And what that means is if they spawn at that night, they can wait and hatch anywhere from 24 to 28 days later, right? Another scenario is they spawn too high. These waves don't reach them. They are forced to wait 24 to 28 days later. So the matching of the spring high tides and the tide height that the parents deposit these eggs is important and can determine if their larvae, their babies, will extend incubation or undergo normal incubation. What's fascinating is there can be a scenario where one group will spawn here, and then another cohort will spawn here, and they both hatch at this given tide. So now you have two cohorts, one group of larval fish that extended incubation, one that went under normal incubation, and now the question is their growth the same? They might have to compete. So how are any changes in growth and development, how can they give the fish an advantage or a disadvantage upon hatching? So what I did was I grew up a bunch of baby grunion, and I forced them to hatch either at 10 days or 28 days. 10 days represented this normal incubation, 28 days represented this extended incubation, and the research question is there growth or development different based on how long they stayed within that egg? So one of the methods that we used was we preserved some of the larvae after we hatched them, whether they were normal incubation or extended incubation. And we did this really interesting clearing and staining process. And I think it's really pretty. It stains the cartilage blue. And it stains any bone that the fish have developed in red. So you can see most of the skeleton, when these larvae hatch, is made of cartilage. There is some bone, but not a lot. To find the bone, we have to look really close. So here we have a picture of a grunion head. Here's its eye socket. Here's its lower jaw. Here's its upper jaw. This is like its cranium here and its backbone. When we take a closer look, I was really interested in feeding, so I was interested in looking at how many of these teeth they had in their lower jaw, their dentary jaw. But what I was more interested is how many teeth they had here in their pharyngeal jaws. So they actually have a second set of jaws here in their throat, and you can see those are the structures that actually developed into bone. They are red, okay? So not a lot of people know that fish have two sets of jaws, and let me demonstrate in this fun cartoon where this sad individual has just been swallowed by a fish how those teeth work. The dentary jaws are used to kind of force that prey into the mouth and kind of keep it there. So if it tries to get out, they'll kind of hook them. But what I find fascinating is the pharyngeal jaws that are in the throat. When that prey gets smashed back there, it literally gets smashed back there, right? So it's literally smashing the prey, and then it'll swallow it, and it'll go to the gut. So fish have these two sets of jaws that do two very different things, and to this day, I swear, whoever came up with this took a fish biology course or was a fish biologist at one point, right? If you're interested in more of this, look up the moray eels, because their pharyngeal jaws are almost as amazing as this alien shown here. All right? So it was my job to process hundreds of these fish, count their teeth. Oh my goodness, it took forever. But anywho, I made it, so can you guys, right? If you guys decide to go into science. And this was their normal, this was the average, right? So the dentary teeth, the average range between around 14.1 for those that went normal incubation, and those that extended incubation had more dentary teeth. Okay, as to be expected, but what was really interesting is they had more pharyngeal jaws, right? More pharyngeal teeth. So the question then became, how do these teeth help these fish capture prey? Does it help them eat them faster, process them faster? So I did another group of experiments where, again, I have to say this, I felt like a kid who put two beetles in a jar, okay? I put fish with a bunch of the rotifers in a jar. I let them wait for 4 hours, and then I counted how many rotifers these things ate, how many of these zooplankton they ate. Okay, but I did this with many of these fish to find out that those that underwent normal incubation on average were eating about 7 of these prey in an hour. Those that extend incubation were eating significantly more of these prey. So I was like, sweet, these teeth are helping them eat. But that's not necessarily true. It could be helping them eat. They are eating more. They do have more teeth as they develop. So originally, these are some of the hypotheses that we help support. Grunion that extend incubation had more of these teeth and eat at greater rates. That was awesome. We were able to determine that these fish, which is really neat, even though one extends incubation and one undergoes normal incubation, they were the same length. So something in their development told them to kind of stop at a given length and kind of wait for them to hatch. So that was really neat. So the other thing that we looked at is that those that extend incubation had more bones, right? They actually had more of these skeletal structures, I call them bones, but they were made of cartilage. And we were interested in, does it make them swim any better? It didn't. They had similar swimming abilities, okay? So again, originally I was like, this increase in teeth for those that extend incubation, that might explain why they have greater rates, feeding rates. I don't know if you see it, but there's a big problem that we were finding. I don't know if you see it in these two, but the big difference is this, right? Fish that underwent normal incubation had more yolk than those that underwent extended incubation, and that makes sense. These group of fish were waiting in the egg to hatch, so they used that yolk for nourishment as they're waiting. So the real question then became, okay, I found all these cool results, awesome. But are these fish simply more hungry? Are these fish here who extend incubation, can they survive even if they hatch? So one of the things we found is if these fish don't eat within about 24 hours after they hatch, they're done. It doesn't matter, right? So they have a very short time to learn to find food after they hatch. In this wave, there's lots of wave action. They have to go find their prey. They're limited on how long they can find food, where these can wait up to 5 days. So this yolk trade-off became a real big problem that honestly, I didn't stick around for my master's project to figure that out. I left it for someone else. Right? It's really neat that these fish have adapted this ability to extend incubation regardless if it's a benefit or not. And that's one of the things that I kept concluding, and I kept saying that it's just an amazing thing that these fish can do this. They can extend incubation, wait 2 spring high tides, and sometimes even 3. But we don't really understand how that affects their abilities to survive in the wild. Okay, that's where I'm going to stop at that project. But one of the conservation things that came up, and when I was learning a lot about grunion, is that human impacts on the beach definitely have been shown to affect when these animals spawn. Too many people in the beach may actually be a deterrent for them to spawn. This happens sometimes when these fish are spawning. I've seen these things come, move the sand, and then you just see a bunch of eggs just lying about and the birds just come in droves to eat them all, right? So some of the human activities may actually be affecting when these fish spawn, forcing them to either undergo normal or extended incubation, which we still don't fully understand. One of the nice things that we've been able to do as a part of continually to study, research this animal, is now there are more limits to when people can collect grunion. So that's good. And that's been a recent thing that there's more, I think now there's 3 months out of the year that you cannot collect grunion. So hopefully that doesn't affect their population as much as these types of activities do. If I thanked everybody who was part of this, I'd be here for a long time. But I just want to make sure that you're all aware that it takes a tribe. It takes a tribe to do research. And my tribe for this project was helped by Cal State Fullerton CMA was huge in this. Again, this is the time I was learning to be a scientist, and a lot of funding support from the Southern California Academy of Sciences. So a major shout out to all of these folks. We're going to focus to our next species of larval fish. This is the zebrafish. Again, freshwater fish. Let's not give it too much grief. But what zebrafish are they're great models for studying the lateral line system. So with the previous project, I was studying how does the skeleton, how does the jaw development, how does swimming abilities allow these fish to eat? In this research project, I was interested, how does their sensory development allow these fish to find food? Zebrafish are a model species studied by a lot of scientists, and they're actually, very similar to goldfish. You can find them at pretty much any pet store. But one of the nice things about these fish is, again, they're a great model, especially study the lateral line system. So for those of us who are unfamiliar with the lateral line system, there are a bunch of these hair cells. I don't know if you see them here. They're glowing like little Christmas tree lights. I actually stained these fish with this stain. It's called DAPSB. And it allows them to glow, those little hair cells that make up the lateral line system. It allows them to glow so we can see them. We can visualize them. Okay? And even the larval zebrafish possess these. So here's the little larval zebrafish, 5 days old, as cute as a little button, right? And all these little Christmas tree lights are its hair cells that make up its lateral line system. So those hair cells are really fascinating because they stick out of the skin, like this one shown here on the lower left. And there's these bundles of hairs. They're normally covered with a little gel-like cupula. We don't see it here because of the type of imaging taken on this, but we do see the hair cells very well. And the way they work is, here's this little gel capsule, right? It's called a cupula. Here are the hair cells sticking out of the fish's skin. It's a little cartoon. And the way they work is when a water force flows right by them, okay, that cupula will bend, and that'll allow the fish to detect these water flows. So this is of great interest to a lot of scientists. How do the fish use these hair cells to interpret what's around them? In this video, let me set this video up real quick. This is a larval zebrafish looked at from the top. You can kind of see its eyes right here, but these little bits, these are actually shrimp in the tank with it. And when I play this, you're going to see this little fish kind of in slow motion. It's going to find one of these shrimp, and he is just going to gobble it up, or she, right? And this fish just sucks it up. Folks, this is in the dark. This fish can detect those little swimming shrimp with its lateral line system and gobble up his food. How do we film this in the dark? We're using infrared light. That's why the resolution isn't super on this camera, right? So they're depending on this system to find food at night. And it's been shown that fish that find food at night are more likely to survive because they're constantly getting that food source. Similar to like feeding babies at night, human babies, right? We have to feed them around the clock. Same thing with larval zebrafish. All right, next slide. Okay, so what I did is I used fish that had their lateral line system, and I put them in tanks here, and I allowed them to just forage. How are they going to perform? So this little fish, this is live. I mean, they're really fast. They're zipping around, all right? And I measured how much of their prey are they capturing based on their age. So when they're really young, they capture really low amounts of prey. As they get older, they become more proficient and they capture more prey as they grow towards becoming a juvenile. But again, I was interested in this lateral line system. So what happens when we knock it out? Well, that's exactly what I did. right? And I use this antibiotic, it's called neomycin. Okay, so neomycin is the antibiotic that's used in Neosporin. You guys are hopefully familiar with Neosporin. It's the same thing that you put on topically, but if you expose these hair cells that make the lateral line system, those hair cells actually die. So this fish effectively has no hair cells, therefore no lateral line system. And my original hypothesis was these fish, even though they're feeding during normal daylight, they're not going to be able to find their prey because this lateral line is important. Well, they proved me wrong. They fed like exactly the same. And I remember I was super bummed out, but my supervisor told me, well, duh, they're using their eyes. I was like, oh, that makes sense. That makes sense. That totally makes sense, right? So that's during the daylight, okay? And then he's like, try it in the dark. So I did the same thing, where I was feeding these larval zebrafish in the dark because we know they can find food in the dark. And folks, this is when it mattered, right? For starters, those with their lateral line, shown in green here, they fed really, really low compared to these shown in green. So their feeding rate was almost cut in half. These are with the lateral line system. Without it, they were poor, poor feeders, right? So yes, the lateral line system matters in the dark. Makes total sense. The lateral line system is key to finding food when vision is no option, okay? But what I was really interested in is, okay, which part of the lateral line system is going to be crucial here? So I got these two data points, fed the fish in the, from feeding the fish in the dark, and here they are again. So this star represents fish that did have the lateral line system. This star shows fish that didn't have their lateral line system. So I was interested, is it only the lateral line system in the head? Because that makes sense, because that's where their mouth is. So I wanted to test that. So I put these fish in blocks of agar, which is like similar to Jello, right? And I cut out just their head so they can breathe, they can be okay. And I only introduced the neomycin just to that little pool. And what happens is their lateral line system in the head is lost, but they still have it in the body. So they still have some ability to detect flow, but just not in the head. And what I found when I did that, they were poor feeders, right? They couldn't, even though they had the lateral line system in their body, it's only absent in the head, they could not feed well. All right, but my advisor's like, that's not enough. Totally makes sense. I understood. It's always a bummer when they say that. Okay. He said, you need to make sure that you putting them in that gel did not mess them up, because putting them in that gel could have stressed them out. And I was like, oh, okay. So I treated them the same way, and then I waited 24 hours. Because they can actually grow the hair cells back after 24 hours. It only kills them temporarily, right? This neomycin. So they were able to grow their hair cells back, they got this treatment, and they were spot on. So they were as equal at feeding as those that were untreated. So the lateral line system in the head is really important for fish to find food in the dark. So we were on to something here. Okay, I was really excited about this. It led to a bunch of other projects. And I'm going to summarize these because these get very, very mathy. Sometimes it gives me a headache. All right. So what we have here is I made this like larval zebrafish lure. And this ball at the end, it's going to vibrate, and you're going to see it move up and down, and it's going to create a water flow similar to those baby shrimp that we feed. Over here, you can see the mouth of a fish, and you're going to, when I play it, it's going to be played in slow motion, so this sphere is going to move really slow up and down, and you're going to see this fish can detect it in the dark. It's It's moving up and down, and this fish, it's already found it. We're just demonstrating that that fish definitely is interested in that piece of glass. It literally was a little bead of glass. Okay, so this was really interesting because now I can control the frequency. I can control how fast this moves up and down and compare how does that look like to other prey sources? How does that look like to shrimp, to worms, etc. So my advisor bought me a fancier camera. That's why it's so much clearer. We were onto something here. So this is feeding in the dark, okay? And it looks so much crisper, but this is in the dark. The fish sees nothing. This is filmed under infrared light. And this rod here, you're gonna see it's vibrating, and this is gonna be in real time. And that fish finds it, and bam. It's gonna play over and over again. I think it's so cool. So I was tricking these fish to bite at glass in the dark, right? But as I changed the frequency, I changed how fast that rod is oscillating. And what we found is that fish respond to higher frequencies. This is when they were the most responsive, at these high frequencies. And that's typically the frequency where larval shrimp, rotifers, what's another one? Copepods, that they are found at. So these fish are looking for known frequencies of waveforms created by typical prey when they're feeding in the dark. But what was really cool to me, and I hope you find this cool, okay? I'm gonna tell you that, because I may be just nerding out here, okay? What was really cool to me is when we followed what's happening with his eye. In this video here, you can see the green outlines the left eye, the red outlines the right eye. And it's going to be played in slow motion. When that fish detects it, that fish's eyes go cross-eyed. I don't know if you saw that there. They kind of stick out. It's going to play over and over again so we can see it. They stick out. This is in the dark, folks. It's still trying to use its eyes to find that prey, right? Its eyes are bending, and what they're literally doing is they're trying to bring their eyes so that they can focus on that prey item. So if there's a prey item here, normally where their eyes overlap are only in that green shaded area. But when they do this eye convergence or divergence, they try to overlap that vision resource, even in the dark, to try to find that vibrating sphere, right? So we did all sorts of really, really neat projects with this, looking at the frequency, looking at how these vibrations cause these elicited responses. But every single time the fish converged its eyes, what I did was I plotted a dot. I had the sphere act as this 0, 0 origin, and that was one data point, for example. I ran a bunch of fish to see where are they responding, how far are they responding from this sphere. And what gets more complicated is they have different types of these hair cells, these lateral line. So those shown in yellow have only the lateral line that are found on the skin. Later in life, they develop a different lateral line system, and what we found was later in life, they can respond further from it. So that gave us a huge piece of evidence that these what we called canal neuromass, these are actually found under the skin, within the bone of the fish. They're actually more sensitive, okay? Without going into some crazy math, yes, adults do sense from their flows from far away, but those neuromass that develop later in life really, really matter. Okay, So for zebrafish depend on vision for hunting. That is well known. That is well understood now. But the lateral line system on their head is what really matters. This is something that we were able to show in the lab, right? No one was crazy enough to think about putting them in Jello until I came along, all right? So we were able to show that it's the head, the system in the head that matters, okay? There's a specific frequency that elicits them to bite. They're like, I want to bite at that food. And these zebrafish are great models for the lateral line responses. I'm not going to go into too much detail, but this is the project that I was working with the Office of Naval Research. They were really interested in how these fish respond to these flows. Because if they extrapolated it and made models and made sensors, they can put these into submarines. And that's what they were really interested in doing, putting these things into submarine so a submarine can respond to a water flow before a pilot could, right? So that was very engineering, more math than I'm comfortable with. I have to tell you this story. I remember going to present this to the engineers and they were really interested. They thought it was really cool. We had to do a lot more math to impress them. But at the end of that, I remember one of the engineers was like, have you ever thought about making a fishing lure to attract these fish? And I was like, what are you talking about? He's like, yes, you have proved using science that these fish are attracted to a specific frequency. Put it in a fishing lure. And I was like, what are you talking about? He's like, if you make a fishing lure, you're backing it by science. And I remember being like, what? And he's like, you're the science. And I was like, I'm the science, you're right. I never went with it, of course, right? I probably made a lot more money if I did, okay? But I do remember, I told one of the audience members here today, I don't know if he recalls, I told him about it, and he was like, sometimes the lure is not to attract the fish. Sometimes the lure is made to attract the fishermen. All you got to do is sell it to them. That's all you have to do. You can back it with science, do it whatever you want. I may have missed my calling, folks, but that is where that engineer's mind went, right? In my mind, the one thing I was thinking about is, man, how are these human impacts changing the dams, changing the water flows of these natural systems? What about dumping? Right? It's well known that these lateral line systems are sensitive to chemicals, sensitive to acidity, alkaline environments, and we're changing these environments like crazy, right? So it's going to affect how baby fish eat. Folks, we affect how baby fish eat, we're going to be affecting how the big fish grow. So that was where my mindset was. Not on making lots of money, clearly, right? But this project was supported by a bunch of resources, bunch of folks. Primarily, I have to thank the folks at UC Irvine and my former advisor, Matt McHenry, and all the folks in his lab, and our funding sources for making this possible. This was only a fraction of one of the 3 chapters of my PhD work, but I figured that this was the most fun to show. Last one. This is the fun one, okay? This one is not so much science, but more conservation. The giant sea bass. Many of us are familiar with the giant sea bass and understand that this is a critically endangered species. These fish get huge. One of the challenges about these fish is they're known for being gentle giants. So unfortunately, that means that they're really not afraid of predators, and that includes us as humans. So because of that, these fish populations have plummeted due to overfishing. Some fascinating facts about these fish is they grow to be over 6 feet in length. Okay, over 600 pounds, easily, max size, of course. Okay, it takes years. These fish will outlive people. So for this fish species to recover, it's going to take a long time. It's going to take multiple generations of people to do something about it, to continuously monitor, to continuously try to understand the species. And even though they get that big, they start off that small. 3 millimeters, folks. 3 millimeters in length. And then they grow up to be over 6 feet long. If you don't understand how much that scales, let me break it down like this. If we as humans grew at that scale, a human baby, average human baby, 6 pounds, 19 inches long, would grow to be taller than the Chrysler building and weigh as much as 8 aircraft carriers. Okay, that's a huge investment that these fish put, in growing. And it can take one person to pull all that investment out in a split of a second. We have to be, very careful and understand the science behind fish populations and how they grow. When I was the research curator here, we were extremely fortunate to work with a group of collaborators that had the giant sea bass spawn in the tanks. Okay? A group of researchers from Cal State Northridge had fish held at the Southern California Marine Institute just across the bridge. And the creme of the Pacific, they said they were up for the task. They called us up and said, hey, let's partner up and try to get these fish to grow in captivity. So some of the staff went out. Many of them are here. I'll acknowledge them here in a bit. And they collected the eggs. So this jar is full of eggs, okay? And I'm going to totally admit, I totally hijacked Eric Dean. Thank you for being here. I hijacked his presentation to tell you about it. So here is the tank that the team set up. You can kind of see these little specks in there. Those are the eggs. They're really tiny, okay? About a day later, they hatched. And to me, I remember looking at this, and I was like, you guys put pepper in the tank. That's what it looked like to me. These are the baby fish. They hatched shortly after, I think within 48 hours for sure, okay? And it was just mind-boggling to see how small they are, how underdeveloped they are, but they were still swimming around. They were, again, just, you guys may not see it, but I think they're super cute. Okay? So the challenge is feeding them. What do we feed them? They're really small. They're really tiny. When they first hatch, they don't even have eyes. They don't even have a mouth. So timing of when we fed them was extremely critical. So if I recall, some of the team stayed and they were looking at these fish. When do the mouths form? When are the mouths the right size? When does the yolk go away? When does their stomach form? And it was just hours of dedication. And I say hours of dedication, and I spent hours upstairs, typing away while they were doing all the work. I supported them to the best that I could, but they did it all. Okay? So their food started off as these zooplankton that they're called rotifers, okay? And rotifers are grown in many, many, many aquaculture facilities to grow baby fish. But one of the downsides to them, first, one of the good sides about having them is they're really relatively easy to maintain in a laboratory setting. The downside is their nutritional value is like popcorn, right? Imagine growing your kids on popcorn, right? That's not enough. It's like potato chips, right? They're just very, very poorly, they just have very, very, very poor nutritional value to them. So the trick was, how do we get them to grow from this size to this size? Because this is when it's important. This early life stage is really important. When to get them to feed, get them into the right size, okay? If you've been into the aquatic nursery, you may see the bubbling green, red, brown, various colors. And these are all algae. The algae are what the rotifers eat, mind you, okay? But what is really, really, really neat is that each different algae possesses different beneficial nutrients. So some of them are really high in carbohydrates, specific carbohydrates. Some of them are high in particular amino acids, omega-3, different types of proteins. Okay? So it's important to get these elements into these plankton. And I don't know if you can kind of see it, but that's their guts. Their guts are full of whatever the team fed them. Okay? And as one of our scientists, I believe Nate is here, he put it best. Oh, just kidding. Hold on. I'm jumping ahead. All right. So these algae are available for us to purchase as paste. And the staff came up with all sorts of different concentrations, looking at which one has the best DHA, which one has the best amount of proteins, and they made this unique concoction, and they named it the sea bass crack. Right? You know, I went with it, they did it, it was up to them, you know, so that was all right. Okay? But this stuff was vital to this success of all of this rearing program. Because as Nate put it, the rotifers are like pasta. Again, imagine feeding your kids just spaghetti, not the best source of nutrients. So they're like ravioli. And those algae is all the stuff you put inside. You can see this nice piece of asparagus there. That's where the nutrition's coming from, right? The nutrition's coming from is from what's inside these rotifers, what we feed them. Their guts are full of cheese and veggies and all the stuff the larval fish need. And then you feed them to them. And then that's how they're getting the most nutrition out of their meal. So this is how we were able to get to this point, getting them to at least 9 days old. It was exciting. I didn't know if it was going to work. I wanted it to work. And then we started seeing, then it got fun. It was fun to begin with. Then it got real fun, okay? Because what happens is as these fish grow, we started noticing their behaviors change. So around this size, you can see they look different. Their colors are different. Their bodies are different. They started settling at the bottom, right? So this is what we call their settlement stage. So they're becoming juveniles. So we were able to start to get them to this juvenile stage. And this was exciting because getting them to this stage meant it was going to be easier for us to feed them. First set that we were able to get them to settle. So they're no longer just floating around, drifting in the ocean. They are settled juveniles. Okay? And then the most amazing part was that they started to display behaviors like their wild counterparts. So I don't know if Mike Kofer is here, if he's listening, major shout out to you because you were a big part of this project. But he took this picture of this juvenile wild giant sea bass and he's just hanging out, or she is just hanging out down by the sand. And what's really neat is when you look at them, they just kind of drift at the bottom. And what they're really doing is they're actually trying to blend in with any drifting seaweeds, any drifting debris at the bottom so they can hide from predators. Right? So we were seeing that in the lab here in the aquatic nursery. That was awesome. And then it got so much fun when they started they just started to beg for food. They're so cute, right? They're so cute. We were able to start feeding them frozen foods. And we were at a point where it was like, they've pretty much made it at this point. They're still pretty small, but I swear in a blink of an eye, we were just overrun with these things. Okay? This is a critically endangered species. We had so many in the aquatic nursery, we had to start sending them everywhere else. And the Aquarium of the Pacific took half of them, thank goodness, because we could not support it in our systems here. It was just too much fish. Endangered species, folks. Okay, it was just too much for us to handle. Look how cute they are. Okay, look at how cute they are. All right, they're really adorable. And one of the things, I don't know if you see it, but they have this beautiful spot pattern here. Okay, this beautiful, beautiful spot pattern. And one of the fascinating things about these fish is this spot pattern is like your it's like your fingerprint. Okay, it sticks with them for life. And we can actually identify them later from photographs. So first we had to celebrate their birthday. All right, let me throw that out there. I should say their hatch day, right? We had to celebrate it. It was a big deal. We were excited. You can see they were excited too, right? So we were able to, not we, Mike Kofer, if you're out there, he took pictures of each and every single fish, the left side, the right side, at this high quality, because there's a program out there, it's called Spotting Giant Sea Bass. And he sent all his pictures to them. He probably crashed their system, I'm not gonna lie to you. But what they do with these pictures is if a fisherman or somebody catches these by accident, they are actually encouraged to take a picture of it and then release it. And then this team uses star constellation softwares on fish to identify them from the pictures that they were taken at, right? So it was really fascinating because he told me that there was at least one hit. There was at least one hit from a fish released here, okay? I don't know if there's gonna be any others, but we ended up with the collaboration, lots of effort, lots of paperwork. The Fish and Wildlife folks probably weren't too fond with me, but I kept sending them paperwork till the last minute. That was on me. But we were able to release over 300 of these little juveniles into Southern California marine waters. It was a huge effort, folks. It takes a tribe. It took a tribe. And it was one of the most amazing projects for me to be on, right? And before I move on, I need to acknowledge the folks that are here today that worked on this project. If you were here when we had the giant sea bass, can you please stand up so we can give you a huge round of applause? I see you guys up front, don't be shy. I'll call you by name. Everyone says I did this. I was in charge of it. I had to deal with all the paperwork and ask for money. But they did all the work, right? Like I said, it takes a tribe. And I couldn't have done it without you guys. So thank you for being part of one of the coolest things that I was a part of here while I was at CMA. Okay, so there's a lot of parties involved. So I'm gonna take the credit. We had the fish here, right? And we were able to send half of them to AOP. We released some, they released some. It was just a wonderful collaboration to be a part of, okay? But in addition to that, we were able to kind of study how they grow. How are they developing? How are these fish changing? How are they undergoing this metamorphosis thing that I began to talk about in the talk earlier today? In addition, we kept some here, and a group from Cal State Northridge and Cal State Long Beach were actually tagging some of them and releasing them into the wild to see where they go. I've kept in contact with some of these folks. They're going in some weird places, unexpected places, and we never knew any of this information. Okay, what's fascinating is that they're using the tracking software that's tracking some white sharks, and our giant sea bass are a part of that. So I'm looking forward to reading that paper one day. I'm hoping that's going to come out soon, or at least an update on that. But that is a to be continued. Okay, so there was lots and lots and lots of folks involved with this giant sea bass project. Okay. Now, a lot of people are like, wow, you're no longer in the aquarium setting because I went to the desert, right? If you're not familiar where the Antelope Valley College is, it is in the desert. It is in Lancaster, okay? When I left my house this morning, it was 34 degrees. It was cold, okay? So I went from one environment to another environment, okay? But I went out there to teach human anatomy and human physiology, because yeah, I'm a fish biologist, why not, right? So how that happened, just to let you know, just so you don't have doubts about my students' education, is I'm a physiologist by training, right? So physiology is very similar between human and fish. You know, there's a book out there, Your Inner Fish, and that's, you know, it's very, very similar. But one of the things that sold me about this campus is this: scorpions, okay? This is a picture I took. You can take a black light out to go scorpion hunting. And what I get to do now is I get to take, hopefully I'm going to be able to take some undergraduates out so that they can learn. This is in their backyard, okay? These animals are in their backyard. But one of the things we don't know about these animals is what is their physiology like. It is greatly misunderstood. Nobody wants to study scorpions. I'll do it, right? I'll do it. So I get to study with our Antelope Valley College crew, and we're starting some projects to study the biochemical mechanism that allows these animals to run, to sprint, for starters, okay? I've done some crazy things at the college. I'm still growing algae out there, okay? I'm still, that's what a picture I took of 2 days ago. I'm still growing algae out there because I'm trying to show students that this is what microalgae is. Students in the desert, you know, I brought some out to a field trip here 2 weeks ago, and none of them had never been here, so it was fantastic. I'm trying to get them, you know, connected to the ocean, because even though they're out there, they influence what happens in the ocean. One of the things I wanted to shout out real quick was our undergraduate research team. They presented a paper. There's the students, list of students' name there. They were tracking, they were presenting a research paper from a researcher at the University of Washington who was tracking eye movement. I almost jumped out of my seat because of my interest in the eye movements of these baby fishes. They were using similar projects, similar research. Major shout out to you guys, did a great job presenting, if any of you guys are hearing. But, you know, some of you guys who are presenting, I hope we do some of this research very, very soon. All right? And that's where I'm going to wrap it up, okay? So I'm going to ask, do you guys have any questions for me regarding any of the 3 projects described today? Thank you for your attention.
Where's the thing? There it is. Thank you so much, Andres. If you're at home and you can hear me hopefully through the computer, I'm a little far away, but if you could enter some questions for Dr. Carrillo into the Q&A, we'll get to those in just a moment. But if there's questions in person, I'll remind you, Andres, if you could please repeat the questions so the folks at home can hear it.
Right there, that man right there.
Chris,
Go ahead.
you know, that one image you have of the zebrafish feeding in the dark. Now, you didn't mention, do they smell?
Good question. So I was asked, the image, the video that I showed of the zebrafish feeding in the dark, the question was, do they smell? The answer is yes, right? But what was really fascinating, even though they can smell, that neomycin treatment only ablated, only removed the lateral, only affected the lateral system. So they're still able to smell, right? And even with the ability to smell, they were poor performers, right? So yes, smell can kind of give them a general, like there's food in front of me, but not pinpoint exactly where it is like the lateral line system could. Right, and we followed up with that. I didn't want to bore you with math and more studies, but yes, that was a good thing that, I'm glad you asked that, because that's something exactly that we followed up on. Yes?
How did you get the sea bass eggs?
How did we get the sea bass eggs? I always want to call the folks in the audience who actually went to go pick them up. So the research group at Cal State Northridge, they were doing a project where they put adult giant sea bass in a tank in a holding facility at Southern California Marine Institute. They were trying to record, because these fish make certain noises, they were trying to record them when they were feeding them, they were trying to record them when they fasted, they were trying to record what noises are they producing. Well, one of the things they started to do was reproduce in the tanks, right? So it was just one of these, like you said, amazing luck, an amazing opportunity we had, and we jumped right on it. We got a call, I sent the team, go get them. Within an hour, the eggs were back here at the aquarium. So it was all a very, very fortunate thing. Yes?
Senor Andres, I know you kept saying that they're so adorable. Were you ever tempted to take one?
I probably shouldn't answer that question. No, just kidding.
I'm just wondering.
So I was asked, I mentioned that they are so adorable. For one, I'm surprised I didn't start baby talking to them during the presentation. Okay, yes, baby fish to me are adorable. Okay, was I tempted to take some of them home? Not because they're adorable. No, just kidding. I was going to say because some people like to eat them. No, I wouldn't do that.
Don't go there.
I wouldn't do that. Not with endangered species. But for me, these images are enough. For me, but yes, I think these fish are absolutely, they're just beautiful, adorable. Even the ones that I did that clearing and staining, I wanted to take some of those home because those fish, I preserved them to show the skeleton. I was so tempted to take one home. My advisor's like, you should take one home. But I decided to leave it in the lab because it could be, someone else can come up with another research question and use those same specimens to answer a different question. So I had to leave it back. I used my best judgment. I regret it a little bit, but that's why I left them back in the lab.
Why don't we do a question from home?
Okay.
Okay. Let's see. Mary D says, you said the giant sea bass that they were released were getting tracked going in very strange places. Where?
I don't know if I'm the authority to say that. So what I can tell you is we released them here in the harbor because the harbor had built some habitat out there. I'm sorry, did that question get sent to the folks?
That was from home, so
Did the folks at home hear me?
I believe they probably, hopefully they heard me.
Okay,
Hopefully they heard
that's what
me.
I'm following
Yeah.
up on. Okay,
Yeah.
sorry, let me continue. So we released them here in the LA Harbor because the LA Harbor, the Port of LA had built some habitat out there that were hopefully going to be used by animals. So one of the things is these receivers are extremely expensive. So we put a tag in the fish, and the fish has to swim by a receiver. Okay, so we were limited to the number of receivers we can put out into the ocean because they could only reach so far. So we decided to put them out in the harbor, put the readers out, and release a few fish at a time. Fish all left. They all left, and that's about all I can say. Some of them have been found like in Redondo Beach, and I know they've probably gone up even more north. That's the last update that I received. So they're moving. They are moving. And everyone always asks me, how do you know something didn't eat them, right? Well, if something ate them, it would have pooped it out and the receiver would be staying at the bottom of the ocean. So it's these little fish that are actually moving and pinging these receivers as they move up and down the coast.
Kind of a follow-up question to that one is from Alice. Is tracking the giant sea bass, can you tell us how large they are now?
I could not. Somebody would have to pull them up on a fishing line, take a picture of them, weigh them, but we could not. When we released them, they had to be pretty much a foot in length, total length, because they had to be the right size. We didn't want to stress them out with the tag, because they got surgically inserted into their abdomen. After it got inserted to the abdomen, they stayed here for some time so we can make sure their behaviors were fine. They were eating within 2 days after the surgery, like nothing happened.
And maybe another question from the audience.
How long does it take them to settle after they hatch?
Well, we were finding that they settled after about 24 to 25 days after hatching. The one thing that I will tell you is ages and everything. What seemed to be more important was the size. So the length that our fish were at 24 days have been generally the smallest they've ever been spotted settled in the ocean. I don't know how old those those are in the ocean that are the same length. So that length, and it was somewhere between 12 to 15 millimeters total length, is when they were starting to settle out, and as big as they've, as small, not as big, as small as they've been found settled in the sandy beach.
Oh, I was really curious, and it was such a great way that you opened about the giant sea bass, saying that they were so big that they weren't afraid. They didn't have predators except us. So I'm assuming they're still endangered. They're still an endangered species. Maybe I don't know if you said that, and maybe I missed it, but how are they going to be protected from us, or will they be if they...
Got it. So the question was, I started off by mentioning that giant sea bass get really big, and that I mentioned that they were gentle giants, right? And at that point, they really have no predators, only really humans. So currently, yes, the giant sea bass are, if you catch one and you bring it and you land it, I mean, you're facing fines and jail time. Okay, and that's in California. Okay, and south of the border, their population's a little different, so they follow slightly different rules. So there are still some fishing allowed of the giant sea bass in Mexico. But here, it was shown that, and this was a mathematical prediction years ago, that there was guesstimated to be less than 200 adult individuals in the wild in Southern California waters. That's really, really low. So again, one of the scary things to me is this fish is so long-lived. It takes them it's going to take the ones we release probably another 10 years, 15 years before they can start reproducing. Right, so we won't see them contributing to the next generation for a long time. So it's going to take years and more efforts, more control, more monitoring. And one of the scariest things for me is, let me, I can't go back this way, sorry, I think I messed this up. One of the scariest things for me is this juvenile does not look like an adult giant sea bass. So an uneducated angler, if they pull this up, they might think, oh, it's a kelp rockfish. And then that fish is out of the population. That is one of the things that scares me. So one of the things that's going to be really important is education. Education on educating the anglers, educating folks, so that we can do our best, to allow this group of fish an opportunity to recover. Yes?
You mentioned that you talked about 40 hours after you were in the nursery. Do they know how long you were?
We don't know exact, because we would get the call.
Repeat the question for the
Oh,
folks
sorry,
at home,
that's
yeah.
right. I mentioned that they hatch within 48 hours after bringing the eggs back to the aquarium. Do we know exactly how long it took them to hatch after fertilization? And that was a question that we wanted to know. Is Kubo in the audience? Kubo is in the audience. Kubo wanted to go down there early morning.
Kubo!
Right? This is Kubo's work right here, this illustration. It's a little blurry, but he is an amazing illustrator. And he wanted to be there early in the morning. I bet you he would have been there at midnight waiting and just waiting and looking to see when these things actually spawn. But we didn't get permission. There was no way we could do that. So we don't know exactly how many hours they had been fertilized by the time the team got there, right? We only know about, how long it took for them to hatch upon the time they got back. So the answer is no, we don't know exactly.
How about just one more question? Dr. Epi.
I was going to ask if everybody here hasn't done it. I think there's one of the ones that was staying here in the kelp tank, right?
There's Jeff.
There's a black sea bass
Yeah.
in that kelp tank. It's up in the right-hand corner, so if you didn't know that or haven't seen it, that's where it is. And they're really, you have to see them to believe them, and they're really, remember, when you look at them, every one of those has individual spots that identify it. So
Is that correct, Jeff? That one's from, okay, so.
Can you repeat that for people at home?
So
They can hear.
people at home, here at the kelp tank in the aquatic, in the exhibit hall, there is a giant sea bass that hatched out, when was that? What year?
2019.
COVID made me lose track of everything. 2019. And it is still here. It is still in the kelp tank. It usually hangs out in the corner. One of the things about them is they have a lot of behavior. If you look at them for hours, if you're a fish person like I am, you can look at them for hours and you can see their little behaviors come out. One of the things I failed to mention is we released these things, where's this? And like a week later, COVID shut everything down. Right, so we didn't really have an opportunity to really celebrate. We didn't have an opportunity to really put everything the news was covering was COVID. We were promised front page by the LA Times. And then
Stupid COVID.
it didn't happen, right? It didn't happen. COVID just completely silenced all of these efforts, which was probably a good thing, because then people weren't out trying to look for these animals, trying to find exactly where we released them. So that's one benefit. But I'm glad that we at least got to acknowledge the team here, because it was all them.
Okay, well, let's have a round of applause for Andres. Stay with us, stay up here, stay up here. What a wonderful talk.